On the 1st of April in 2013, the front cover of Time magazine was adorned with a bold proclamation: how to cure cancer. Yes, it's now possible. Notice this wasn't an April Fool's joke, and while the accompanying article was more nuanced than this headline, the damage was done. Cancer is not one single disease, but hundreds of diseases, each with different genetic origins and treatment strategies. Researchers and clinicians already struggle to manage the expectations of funding bodies, not to mention cancer patients and their families. And headlines like these trivialize the complexity of our work and makes it harder for scientists to do our jobs. If we take a moment though, to consider the general public's perspective, there doesn't appear to be a lack of high-profile scientific projects that are at least in part funded by the taxpayer. Notably the Human Genome Project, involving an international consortium of scientists across 20 countries, and it took over a decade and $3 billion to complete. It aimed to sequence a reference human genome from individual donors and promised to crack the human genetic code and unlock cures to all sorts of diseases, including cancer. It was an incredible achievement in the historical context of biology, and now we somewhat take it for granted given that we can Google the DNA sequence of any gene you can think of. However, unless you are working in the field of genetics or molecular biology, it can be difficult to appreciate this impact on your everyday life. What's the disconnect here? And why are advancements in medical research not happening at the rate we were promised? Hello, if it's your first time here, my name is Jack Wang, and I'm a microbiologist and science educator based in Australia. The Human Genome Project had a transformative impact on health and molecular biosciences, but it can be difficult to appreciate this without understanding the underlying principles. In today's video, we're going to walk through the basics of DNA sequencing with specific emphasis on traditional sequencing, also known as Sanger sequencing. This is the approach they took in the Human Genome Project, and it is still routinely carried out in DNA sequencing facilities all across the world. The field has moved on from this in many respects, and we will also highlight some points of comparison to next generation or next gen DNA sequencing platforms that have taken over the industry. If you imagine the 4 chemical bases of DNA, A, T, G, and C, as letters within our genetic alphabet. They can be assembled in different orders to form words, sentences, or genes. DNA is double-stranded, and each strand serves as the other's mirror image, or reverse complementary sequence. The 2 strands are held together in double helical conformation with hydrogen bonding. A binds to T with 2 hydrogen bonds, and G binds to C with 3 hydrogen bonds. The order or sequence of the bases ultimately dictates the gene's function, and we can use Sanger sequencing to figure this out. Like many techniques in molecular biology, at the heart of Sanger sequencing is a PCR reaction. We can design a PCR primer complementary to the sequence immediately before or upstream of our mystery unknown DNA to initiate the reaction. We want the primer to be 20 to 30 bases in length, have a G plus C percentage of 40 to 60%, a melting temperature or TM between 55 to 65 degrees Celsius, and together these principles make sure that the primer is long enough to bind to the template DNA, that this binding is stable enough with enough G-C combinations, and that the primers can melt and separate into single-stranded DNA at a temperature that also supports its annealing to the template DNA during PCR. This might sound complicated, but there are many online tools that can help design primers very quickly. In a normal PCR reaction, we want to amplify double-stranded DNA, so we design both a forward and a reverse primer to amplify both strands. For Sanger sequencing, we only need 1 primer per reaction, along with the template DNA, DNA polymerase, reaction buffer, and nucleotides. As the sequencing primer anneals to the template DNA, DNA polymerase will bind to the primer and initiate DNA replication using the template DNA as its template. If there is an A in the template strand, it will copy a T. If there is a G in the template, then a C will be copied, and it will continue on like this. Or will it? DNA polymerase can only proceed with this copying if it has the right A, T, G, and C nucleotides available in its immediate surroundings. Within cells, this isn't an issue unless we're very malnourished or lacking specific metabolites. We should have all of these nucleotides or dNTPs in ready supply. Sequencing reactions, though, happen within a test tube, and we control what DNA polymerase has access to. So yes, we do provide dNTPs, the ATP, the TTP, the GTP, and the CTP as part of the reaction, but we also include a dideoxy version of each of them. What does this mean? A dideoxy ATP, for example, has 1 fewer oxygen than the normal dATP, and this oxygen is removed from the 3' hydroxyl group used by DNA polymerase to form a phosphodiester bond with the next nucleotide in the sequence. If a normal dNTP is inserted, the presence of the 3' hydroxyl group will allow DNA synthesis to continue on to the next base. However, if a dideoxynucleotide is inserted, it lacks this 3' hydroxyl group, so DNA polymerase cannot continue and the reaction is terminated. We can control how often the dideoxy versions of these dNTPs are inserted by controlling the relative concentration of dideoxy versus normal dNTPs in the reaction. Let's look at an example reaction with dideoxy ATP. DNA polymerase can bind to the template DNA from the primer and synthesize new DNA. Checks the template, and in this case, it needs to add a T. There is dTTP in the reaction mix, so we can do this. Next is G. Yes, there's dGTP in the mix. And then next, C. Yes, there is dCTP as well. When it needs to add an A, and it has two options, either a dATP and the reaction can continue, or a dideoxy ATP and the reaction stops. Every time an A needs to be added, it's a random choice between dATP or dideoxy ATP. And as the reaction proceeds, there'll be many fragments of DNA with different sizes, as each time a dideoxy ATP is added, the chain is terminated. And this can happen at any point in the sequence where an A should be. When we separate these DNA fragments by size using gel electrophoresis, we can calculate how long each of these bands are. What all of them have in common though is that the last base added should be an A. We know the size of the bands, say 200 nucleotides, and that the last base added is an A. So we can infer the 200th base in this DNA sequence is an A. For T, G, and C, we have separate reactions with dideoxy TTP, GTP, and CTP respectively, and in those reactions, every fragment will end with a T or G or C. When you consider all four reactions, you can put the pieces of the puzzle together and figure out the DNA sequence base by base. One Sanger sequencing reaction can accurately read out about 700 bases. So if your gene is longer than 700 bases, you need to run multiple overlapping sequencing reactions across that stretch of DNA. In fact, to maintain accuracy and fidelity of the output, often a single stretch of DNA sequenced multiple times just to make sure we had the right result. Sanger sequencing machines can run up to 384 samples at a time and sequence 1 million bases per day. That might sound like a lot, but for reference, the human genome is 6 billion bases. So still a ways to go until you can have your genome sequenced at the doctor's office while waiting for a coffee. What gets us closer to that reality is next generation DNA sequencing, which has the potential to run 13 billion samples at a time and sequence 4 trillion bases per day. That's a lot more zeros and orders of magnitude higher in processing power than Sanger sequencing. There are different competing platforms in next-gen sequencing, and comparing them is a topic for another video. It is worth considering though, on a holistic level, how all of these platforms were able to increase their sequencing throughput. Instead of free-floating within a reaction mix, the template DNA can be organized in a consistent and reproducible way, which speeds up the readout. Adapter and capture sequences can be added to either end of the template DNA, which will allow it to bind to microscopic surfaces within a flow cell chamber that forms a uniform matrix to be loaded into the sequencer. Cleaning up the DNA after a sequencing reaction has also never been easier. You can even use magnetic beads in some systems for the nucleic acids to bind to. Also, instead of running 4 separate reactions with the dideoxy versions of A, T, G, or C, we can use fluorescently tagged dideoxy nucleotides instead, where each different base fluoresces a different color, which can be read by a laser detector. This way we can combine all 4 dideoxy reactions into one tube. Instead of running the samples on an old school agarose gel, we can use capillary gel electrophoresis instead, which passes an electric current through the tube, which then organizes the DNA bands by size. As the bands flow through one by one, from smallest to largest, the fluorescent tag on the dideoxy terminated nucleotide will be imaged by the laser detector, and the sequence is automatically built. Each of these efficiencies also makes them more accessible for multiplexing, using liquid handling robotics to deal with hundreds and thousands of wells across multiple plates. Now that the cost of DNA sequencing has come down dramatically, how close are we to realizing the potential of the original Human Genome project? With the benefit of hindsight, we know that there's still a big gap between knowing a gene's DNA sequence and being able to predict or change its function within a human being. How is the gene made into protein, and how much protein is needed for the process to function properly? Do multiple genes perform the same function, and are there redundancies built in if any of them stop working? How many variations of the same gene exist across a human population, and what impact do these variations have? The original Human Genome Project sequenced DNA from only a handful of individual donors, and really we need to sequence millions of humans to get a better sense of the genetic basis of diseases and the inherent variation within our species. There's a lot of nuance and complexity in biology, which is often difficult to relay to the general public. It is up to scientists to bridge this gap and communicate the significance of our work at every turn. This is the Biolab Collective. I'm Jack Wayne, and I'll see you in the next video.